to this computer. Okay. All right. I'd like to welcome everyone to our first WISE seminar of this spring semester. We have two events planned for this semester thus far. And so today, of course, is the first of those two. I will give Dr. Matos a brief introduction before I let her kind of take over. Dr. Mayara Matos is currently a senior staff fellow biologist in the Center for Food Safety and Applied Nutrition at the U.S. Food and Drug Administration. Prior to this appointment at the FDA, she received her master's in biochemistry from the Federal University of Ceará, Brazil, and then her PhD in biology from West Virginia University. She then completed two postdoctoral appointments, one with the FDA and the other with the University of Maryland. Throughout her career, as Dr. Matos will detail today, she's been involved in several interdisciplinary projects, including research in plant biochemistry, pharmacology, forensics, and food science. Her current research interests focus on the development of analytical methods to address safety issues in seafood, such as the identification and quantification of metabolites that indicate seafood decomposition. Dr. Matos will further detail her professional journey to the FDA today, as well as some of that research that I just mentioned. So I would like to officially welcome Dr. Matos and thank her very much for taking time from her schedule to join us and be that first invited speaker of this semester. And before I do hand things over to her, I will mention to the students in the audience, first of all, you don't have to tell me that you're attending. I will get the attendance through Zoom. Okay, so don't worry about that. And I will email it to the appropriate professors. If you have questions throughout the lecture, you feel free to put them into the chat box. If they're pertinent to what Dr. Matos is discussing at the moment, I'll interrupt her to ask that question. Otherwise, we'll save the questions for after her talk, where we'll have a short Q&A. Okay, so with that, let me hand things over to Dr. Matos, and she can share her slides.
First of all, thank you very much for the introduction, Dr. Haley. I appreciate the invitation to give this talk today at the William Paterson University. I was telling you earlier, it'll be awesome if I could have been in person, but unfortunately that didn't happen, but perhaps in the future. So let me just share my screen. Make sure everybody's in here. Can you see my screen?
Yes, we can.
Yes, okay, perfect. All right, so once again, hello everyone. Thank you for attending this talk today. I think it's a great initiative from the university to promote seminars like this, where students can learn about the speakers beyond their work. I think usually seminars that only show research are super valuable. It can be awesome. But it's only when we know that person's journey to where they are right now is that when you can understand that even though we might be in different stages in our careers, we all probably had very similar doubts throughout the way. And that's always very valuable. I wish I had had that experience when I was a student. But today I'm presenting a little bit of my current research and a little bit of my previous research along with my journey. I try to focus a little bit more on the journey itself, but you see in each stage how that happened. And as my title says, and also the different pictures here, you hear that I had a very non-linear career in research. And I hope that with my story, I can inspire you to continue to pursue your goals, even if you maybe right now you are not sure if they are very clear for you. But let's see. Let's go to it then. Because I'm an employee at the FDA, I have to present this disclaimer slide where this presentation reflects the views of the speaker, in this case mine, of the authors, and does not necessarily reflect those of the U.S. Food and Drug Administration, and any mention of commercial products is for clarification only and is not intended as approval, endorsement, or recommendation. Having this disclaimer done, let's move on. And again, so here's my journey pretty much. Even though my research career was non-linear, I still followed the regular academic path to obtain my degrees. So as Dr. Haley presented in her introduction, I hold a bachelor's and a master's degree from, actually, come back. I hold a bachelor's degree and a licentiate degree in biology. In this case, a licentiate degree, if you have never heard, is it's something, it's the same length as a bachelor's, but it's a focus in education, a specific scientific field. In my case, it was in biology. And then I got my master's in biochemistry with emphasis in plant biochemistry. All of these from the Federal University of Ceará, or Universidade Federal do Ceará. And then I received a PhD in biology with emphasis in forensics and analytical chemistry from West Virginia University, followed by 2 postdoctoral appointments where I worked with food safety applications, and one of them was with the FDA, and the other one was with the University of Maryland. The University of Maryland has a joint program with the FDA where we can do a postdoc, but at the same time work at FDA facilities, and the projects are very correlated. And then how all that worked out so that I now have my current position as a biologist at the FDA. Well, as you may have noticed from not only from the university that I mentioned, but also a little bit from my accent, I'm not from the US. I'm actually from Brazil. And I'm from this state, highlighted here, Ceará, and my hometown is Fortaleza. This is a city by the coast, very beautiful city. As you can see by the map, we are very near the equator, so we have summer all year long. The temperatures are about 88 to 90 degrees all year long. And when you have a rainy season, which is for us considered cold, we might reach around 75 degrees Fahrenheit, and that's very cold. People use long jackets and all that, and it's like it's very funny when I moved to the US to see the difference in temperatures. Yeah, so it's a very nice city over there. Population-wise, somewhat a little more populated than Houston, Texas, a little less than Chicago, so it's very nice. The state itself has very different types of landscapes, very beautiful, different types of beaches and mountains, semi-arid areas, lakes. So it's all a lot of nature around that. And it was growing up in this state and always watching all those TV shows, kind of similar to National Geographic ones about natures and animals from Brazil and all that. That I, since a very small kid, I was always interested in becoming a biologist. So like, all right, so I would like biology. I like all that. Let me enroll in the biology course in the university, and that's how I in my family, and that started the first chapter in my career by joining the Universidade Federal do Ceará, or as the abbreviation shows, as UFC, not the Ultimate Fighting Championship, although I appreciate the martial arts, but I usually have to spell it out, otherwise people think it's something else. And then over there, I work with natural products and plant biochemistry and all that. And there was a strong focus on medicinal plants that were native from Brazil and from the region, and how the population used specific types of plants for different medicinal purposes. So when I joined the university, I pretty much very soon, I realized that even though I liked all those animal shows, I was not very fit for the animal research and going to field and all that. I actually enjoy much more the research in the lab and knowing how things work in a smaller level. So that's how I joined more research related to biochemistry. And my interest was pretty much carried by this specific plant. So this is a plant, is the species Montanus drasticus, is a plant that is native from the northeast region of Brazil. And it's very used in a popular medicine over there for the cure of pretty much everything. Cancer, inflammation, all sort of things. And the reason why I was very interested in this plant specifically, and actually when I entered in research, I wanted only to study this one, was because of a family case that I had, where my grandmother, before I was born, she was diagnosed with colon cancer. And the doctors, when they gave her the description that the level of cancer that she had, it was already too late to have any treatment or any chemotherapy. So they gave her 6 months to live. They said, there is no treatment that you can do anymore. Go home, enjoy your life for whatever time that you have left. So she did. And when she came back, one of the neighbors, I believe, said, why don't you get one of these bottles? They are sold from the market. And these bottles are called milk of Janaguba. Janaguba is the popular name for this plant. And what this milk is a latex. So this plant, from the stem of this plant, you can remove this latex. Latex is the same compound that is used to produce rubber or any rubber-like materials. So they remove this latex from the plant and they dilute with water and they put in these bottles and they sell in the popular markets along with other herbs and flowers and all that. And people may use that for medicinal purposes. So a neighbor gave it to her and said, well, we have nothing to lose. You have cancer, take that. Well, a year passed. She came back to her doctor and he was highly surprised that she was, first of all, alive. And then, well, what happened? How you were here after? You should be long gone. Well, he did all the exams again, and she had no cancer anymore. My grandmother happened to live until she was 97, and she was super healthy. She passed from something else that was not colon cancer. So knowing that story, when I entered the university, I want to do research with this specific plant. I found a lab that did research, and the way we did was we would separate using different type of separation techniques, such as chromatography. We would separate the macromolecules that we thought it would be interesting and could have a potential medicinal applications. And with that, that was my main interest. But I didn't work only with that. I worked with other types of plants, and these are some of the ones that I work with. Some of them also had latex. This one is the latex on the leaves, and it has pretty much an entire plant. And this is there's flowers and some seeds. And the interesting case about this one called Calotropis procera, this is the type of, it's pretty much a bush that you can find anywhere. It's not native from Brazil. You can find it in different countries that tropical climate. And people use this, the latex from this plant, if they have a mole, if they have any type of skin rash, and they can use it. And it's pretty effective against those type of problems. At the same time, if you try to drink the latex from this plant, different from the first one that I presented, it's highly toxic and you can die really quickly with a very small concentration. So it was very interesting that when I'm working with this plant, we could separate which fractions within the latex and which compounds were responsible for each one of the properties. In this case, we were working mainly with proteins and a few secondary metabolites, but that was very interesting. And the same likewise with some flowers and some seeds, what you could extract from them that had those properties. And a combination of results that we have, in this case for these three plants, is that they showed very high anti-inflammatory properties, anti-tumor properties, analgesic, and even control of mosquitoes, which some of those latexes we could use with the eggs of the mosquito transmitter of dengue, and they would stop the developing of the larvae, and in this way we could eventually use for the control of the mosquito population. And tying back to my main interest, which was the plant that cured my grandmother actually has these properties or not, and figure out that one of the results is that yes, it did have. We collaborated with some of the medical doctors that also happen to be researchers in the university, and they had similar cases with their own patients where they diagnosed cancer in their patients, and they took some of these medicinal plants, and eventually they were just somehow cured. So they were highly interested in this type of research as well. And that plant specifically did have anti-cancer and high anti-inflammatory properties. So that was very interesting. I was really excited when I did that. And I did that throughout my undergrad and master's. And it was always very nice and everything. But despite the great results that I was having, I I was not very happy with the research group that I was working with. And I was always thinking, I like biology and I like these projects that I'm working on, but I don't know if that's all I want to do for the rest of my career. I also like other things in biology, other topics. And at that time, one thing that I was very interested on was in forensics. Like, oh, how can I use the knowledge that I've gained uh, for the research that I've been doing my undergrad and master's, like towards something else as a project. So I was thinking, well, maybe I want to change a field and I want to see if I can use that towards forensics. So at that time, Brazil had just initiated a program to boost Brazilian science and technology through the expansion of international mobility, which means that the government would start this program and you'd finance students of any universities if we could apply and you could send a project and they would see if that was valid and then they would pay for your studies abroad. So I applied for PhD in the US and I passed through a competition nationwide and they selected a few people and fortunately I was one of them. And this program was very important because it was probably one of the very first times in Brazil where if students from less income families like mine had the opportunity to compete equally with all the students. Usually these opportunities of studying abroad or even studying in another state in the country were only possible for students who have more financial means so that their families could sponsor that. But mine definitely didn't have that. I would not be able to even move to another state if it was from my own family income. So this was very important that I had the opportunity to come abroad. So once that was established and I competed for that and everything, had to learn what GRE was, what TOEFL was. Like this, the requests are very different from what we have in Brazil. And I talked to my advisor and actually throughout the process, not only after the result. And the response was very different than what one would expect. He actually didn't give any support at all. He thought that I was a dreamer, that this was not possible, that I should leave this type of adventure to people who had more means and were more used to that. How would I go and go to another country where I didn't know anybody and didn't know, he thought anything about the language or anything and he start over. He's like, that will never work and I'll not give any support. So he just washed his hands and said, I'm doing nothing. And I heard the same from a few other professors. And I was like, wow, I could have used that as a demotivational speech, but instead I use that as a fuel to say, that's what I want. And if you think I cannot do, that's one more reason to prove you wrong. And I got recommendation letters from different professors, and they helped. And that's how I moved from Brazil to the US and to start a new chapter in my career. That was when I applied to do my PhD, and that was in West Virginia University, where I started working with forensic in a very nice group, very supportive, in a very different atmosphere than what I had. Not saying that my previous group was not supportive. My lab coworkers were always very supportive. It was my advisor at the time, which happens. Sometimes we encounter all different types of people. Sometimes we expect some of them to be more supportive, but they are not, but that's okay. Can always overcome those issues. So in West Virginia University, new chapter, everything very different. First of all, moving to the US, I had no family or friends here, didn't know anybody. It was not only a different country, different language, different food, different everything. One very noticeable thing for me was the weather. As I told you before, I came from a place where it was about 90 degrees all year long. And I went to West Virginia where it was cold. It was the first time I saw snow. It was very different landscape. Brazil is very well known for its soccer. There was no soccer, it was all American football. Everybody was very enthusiastic about the Mountaineers and football, and I had to learn about that as well. But one thing I really liked was being exposed to different cultures, not only the American culture, but also people from all over the place. I could learn a lot about them and they all the experiences as well, the life story. And as I said, a new research group, very welcoming, very supportive. And over there, I started working with this technique. It's called isotope ratio mass spectrometry. This technique has been, I'm there to say, a very indispensable tool in several disciplines, and especially in forensic science, because it has the ability to unravel the source of chemically indistinguishable substances, which means that, for example, you have two compounds or two different types of, let's say, drugs, and chemically speaking, they are exactly the same. You have no idea, based on different analytical techniques, if one came from one source or the other. But this technique uses the measurements of stable isotopes. So when you think about isotopes, you might remember carbon-14, the radioactive ones, but our bodies and pretty much everything in the world is composed by isotopes, most of them stable. And those are carbon-13, you have nitrogen, you have sulfur, all the ones that do not decay. And everything that is composed by those isotopes, you can trace them back and figure out where their source is from. So that's why it's this technique can be applied in forensics, because it can help you with human provenance. We can help with wildlife forensics, food authentication, pollution. Is this compound coming from source A or B? Is this ink coming from this location of the second one? Explosive fibers, and so on and so forth. So it's a very versatile technique in forensics and other disciplines as well, but I was more interested in the forensic application. So one of the projects that I work with was with human hair. And you might have heard the expression that we are what we eat. And that's true most of the case. There are a few caveats over there, but let's keep on the overall saying that we are what we eat. And the reason for that is the isotopic composition of our body tissues come from all that we consumed. So everything is made of carbon, nitrogen, sulfur, hydrogen, oxygen, and everything we consume is reflected in body tissue. So we can know, based on that, a few biometric traits from a person that maybe you couldn't figure out just if you didn't have DNA, of the DNA was degraded. So one of my researchers with human hair, so let's say that you get strands of hair and you don't have a DNA that's just on the root, or the DNA is degraded. What can you tell about that individual just based on the isotopic composition of that hair? And we could find several things. You could identify if a person, based on a survey of several volunteers from different groups and age ranges and diets and all that, which people were omnivorous, which ones ate meat, which ones were vegan, which ones were vegetarian. In terms of age range, we could differentiate if they were infants, or if they were older, if they were about maybe say 27 years apart or so, we would be able to differentiate them. You could say if the person was had more alcohol consumption than others, and all that was based on the isotopic composition of, in this case, we did from the amino acids that were extracted from the hair. And what could you tell from that research? Well, that provides a lot of investigative leads in forensic work, because if you don't have any DNA sample that might be helpful, of course, DNA is always the golden type of analysis in forensics. But if you don't have that, you need something else to support that. Any type of information that can not exactly pinpoint a person, because that's usually DNA's work, but that you can remove someone from the pool of suspects, that is a great investigative lead and can be very helpful. So this was one of the projects. Another one that I worked with was more with forensic entomology. And in this case, was also used the same technique, but using a few other things, elemental analysis, liquid chromatography. And this one, we work with blowflies. If you have ever seen that subject animal, or hopefully not a person, but adult blowflies, one of the first necrophagous insects that arrive at a fresh carcass, and they lay their eggs in the flesh, and then those eggs will hatch into larvae, and that larvae will feed on the decomposing body. And as I mentioned before, like all organisms, flies are also what they eat. So when we analyze the isotopic composition of those flies, of the larvae, of the pupae of the adult fly, we can track back to the fly diet, to the original, to the what they ate initially. So in a combination of ecology and also forensics, with this research, we wanted to um, enhance the significance of blowflies for postmortem interval investigations, because you could use, in this case, we investigated what if these flies laid eggs in different types of flesh, let's say beef, chicken, and pork, and we carry on the analysis throughout the life cycle of the fly until they became an adult, the eggs to the adult. And then we were able to differentiate from which sources those larvae initially had fed on, and then eventually they had the signature on the adult fly. And we could have a few other information ecology-wise, just about the biology of the fly and all that. But the interesting thing is, we could, by knowing what the fly had fed on, in this case, we didn't use any human flesh, but that could be used in forensic applications for humans in situations such as tragic situations where people do not have access to victims. They are under some areas, but they do have access to the necrophagous insects that are going over there. They can figure out, based on the difference in diets, if that fly was feeding on a human body or a deceased body of an animal or something else, and then eventually be helpful with those postmortem investigations, postmortem interval determination. So that was also a very interesting type of research. And it's still the forensic wise, totally different, not more forensic knowledge, but now food safety. I had some projects involving not only West Virginia University, but also the Florida Department of Agriculture and Consumer Services, and the FDA as well, where we were working with oysters. And the reason why we were interested in oysters is because oysters, you might know, play a key ecological role in stewarding food web. They maintain all the nursery ecosystem, they filter everything, and they lead to a greater diversity in the aquatic species. And not only in terms of the ecology of the oysters and the ecological benefits, but the economic cost. If you ever go to a restaurant and you try to order oysters, you might see that they are very expensive, very, very expensive. And some groups try to sell their specific oysters. They are sold as boutique oysters. They have a higher cost depending where they are coming from, depending on the minerals of the area and all that. So how can we know if those oysters are actually coming, they are the same species? How can we know if they're actually coming from a region that is considered better and in terms of minerals, and because of that they could be sold more higher cost, or if they are coming from an area that should be closed for reproduction period and they are actually not? How can you tell from that if DNA is the same because they are the same species, or if you don't have any GPS tracking available because of whatever reasons? We could also use isotope analysis for that. And what we did was we had some oysters from different areas, and here I'm showing just part of some of the results we have. We had oysters come from adjacent areas in the Florida Bay here. They're the same species, but these bays were coming from, they had different freshwater inputs. And when you have different water inputs, the oysters might be exposed to different anthropogenic contaminations. So some areas might be more polluted than others. And we were able to differentiate oysters that came even with very closely related neighboring regions if they were coming from one or the other. So our ability to discriminate these adjacent areas had a lot of impact for not only consumer protection, because then you can say that if whoever's selling those oysters, we are protecting the consumers and knowing that exactly they're coming from where the source is saying that they are coming from, but also public health, human public health. You were saying that those oysters are coming from areas they are clean, they are open for harvest, that they are not contaminated with, let's say, Vibrio, and might causing high food contamination. So there was also very interesting results. And it was actually this research here with food authentication that made a bridge to what I do in the FDA. when I was still in my doing my PhD, I applied for internships at the FDA, and I did 2 summer internships here, working with seafood and a few other projects on the side, but mainly seafood. And after that, it was based on the work that I did during those internships that I was offered a postdoc, and I continue working in the same area. And that started the next chapter. That's when I started working for the FDA, U.S. Food and Drug Administration, more towards food safety. And you might think, well, your other projects were pretty nice, and I agree. I really like working with forensics. But when I saw the different applications that I could have, like, well, I also like a lot of public health stuff in food. Can also apply what I learned before in this field, and I knew that I could. So why not try this different avenue and type of research and learn something new? So that's what I did. And that's when I joined the Center for Food Safety and Applied Nutrition. That's also called CFSUN. The CFSUN is one of the centers from the FDA, and it provides services to consumers, domestic and foreign industry, and other groups, and has a lot of scientific analysis and support other than the regular FDA agency administrative tasks. But it is responsible for a lot of the planning and handling of critical issues related to food, dietary supplements, and cosmetics. So within these 3 fields, I work with the food side of the research specifically. And with that, what the center does and what the FDA helps is to safeguard that the food supply that we have in the US. And what we do, the way it's done is by evaluating the use of chemicals as food ingredients, and also substances that may come into contact with the food. Is, for example, when you have carry out from a restaurant, when you buy some fast food, that packaging that comes through, we want to know if there is any sort of compound migration that comes from the package, the food, that might be harmful. And then the FDA monitor the food supply for any chemical contaminants, and you take action if you find that there is any contaminant that might be unsafe for consumption. And all that information, if you check the FDA website, you might see the same, almost quote-wise what I said about the type of research. So the type of research that I did there, doing my both postdocs with UMD, and the first postdoc with FDA and the second with UMD, with kind of an extension of each other, of the other one, is with seafood. So when seafood comes from the US, the majority of food that's consumed in the US come from abroad, from different places. They come into the ports, and there has to be some sort of initial screening and evaluation to see if that seafood is actually good. And currently, the FDA relies a lot on sensory or organoleptic analysis, which means that someone who is highly trained, and don't be mistaken that, oh, it's just some person just smells a fish and it passes. Like, no, those are people that have years of training of in a way that they can smell it, they can give scores and differentiate if something is decomposed, if it's not, if it can pass, or if it needs further analysis, if it's something that's being suspected. It's a very hard work. I don't think I would be able to do that type of job. So I really admire people that work on the sensory analysis. Is a hard job that they do. And they do that, the sensory analysis, and they have a few other analyses, such as a few compounds is histamine, indole, which is for seafood specifically. Other ones, mainly histamine, that can cause seafood poisoning. Sconboy toxin poisoning is the ones that sometimes you might hear on the news. There's specific types of seafood that people eat and they get very sick, and in an extreme case that might cause death. That might be from high concentrations of certain compounds. They are related to decomposing seafood. Even if you cannot really smell it or see based on the appearance of the fish, you can still determine if they are present or not. And my work is combining analytical techniques to support that sensory analysis and make the work of the field labs and the regulatory side of the agency more effective. So one of the projects that I worked on is, for example, we got several species of different types of fish, and they were analyzed by the sensory team, and they had several levels of decomposition. So some of them were considered super fresh, because the ones that we usually eat, and some of them were a little bit here and there, so like kind of what we call the borderline path. This is almost getting bad, and this could be almost good, but has passed a little bit. And the ones who are highly decomposed that you could immediately see they were bad. So my work was to create the methods, analytical methods, and focus on a few compounds. In this case here, I'm highlighting some: putrescine, cadaverine, histamine, tyramine. Some of them you might feel like putrescine, cadaverine. They are actually compounds that were initially extracted from these human bodies, that's cadaverine, and there was the one that gives a very putrid and rancid smell to any food, pretty much, and in this case, seafood as well. And the other one, histamine, as I said, is the one that is highly responsible for spermboid fish duct poisoning. So what we did was we would try to identify which species had any of these compounds and quantify them, see how much it was present, if there was hazardous for consumption or not. We will apply some statistical analysis, and we'll be able to differentiate the decomposition stages, even the ones that were very closely related to each other in terms of decomposition. So you might see that I did all sort of different projects. Each one of them could have been a career itself. Some people have their entire life just working with food safety and food science. Some of them just forensics, some of them just plant natural products, but I worked a little bit with each one of them. And in this non-linear career path, the advantage that I see is that you gain a lot of knowledge across the different topics. As I said, you always learn a little bit. It's like I like biology as a general, and the interdisciplinary that it can have with different disciplines, such as chemistry, physics, and statistics, all of that. So I like all that interdisciplinary. I didn't want to be doing just the same thing all the time. Another advantage that I think is very important is that you become adaptable to new conditions very quick, because every time you change, you have to catch up with the knowledge. You have to learn what is the current techniques that are being used. What can I apply that is different that I didn't use before? So you become very adaptable. Also, you get a broader networking, because you are across your journey, you are getting contacts from each one of those fields. So in the future, if you want to make some interdisciplinary project, or you want to return to that field that you used to work on, you can always contact some of those people that recruit throughout your career. And I think the biggest advantage, at least for me, it's just interesting. I find it very exciting every time I see a new project like this that's very different from what I do. Like, yeah, I want to learn that. I want to work with that. So it's, for me, it's an advantage. But not everything is always on the good side. And I have to be honest, there are some challenges. And for me, is that the challenge that you always have to be out of your comfort zone. When you are getting used to one technique and you always see the same thing, you are doing the same, you get used to the same type of background papers. And then when you switch, it's a new field. You are relearning everything. And that can be a little uncomfortable sometimes, being out of your comfort zone. Also, because you're always having to be out of your comfort zone, you have to work harder to learn the new subjects. You enter a new field, and you might use some techniques and some knowledge that you had previously, but you have to continue. It's a continual learning. It's a continuing learning. You have to keep working hard to catch up with that. If your plan is to work in academia, that might be a little hard to adjust, just because, for example, as a student, if you want to join the research group of a professor, you pretty much want someone who is consistent and it has worked throughout the entire career. You don't want someone that, for example, you are interested in microbiology and the professor has a microbiology research group, and suddenly, oh, you know what? I want to switch to something to ecology, not related to micro at all. So that might be challenging. But if you are able to combine and make your research interdisciplinary, that is highly rewarding, because then you expand the scope of your research, you expand the scope of what types of grants can be achieved, what type of students you can get, and you create projects and results that are beyond what is expected from the field. So those are very interesting ones if you are interested in continuing academia. Another challenge, and that might be not only this non-linear career path, but this might be one of the biggest ones, is the imposter syndrome. Keep thinking, if I'm switching all the time, like, can I do that? Will people trust me if I keep switching? Will they trust my knowledge that I learned all the way? So that's something that you have to keep working on, to trust that yes, you can do it. And it's part of some of the questions that I had. And maybe if you're thinking on the same lines as I did, like, what are the questions that in an early career path? Can I do it? Will I get a job if I continue, if I keep changing? Will people trust my knowledge if I didn't continue in the same subject route since undergrad? And can I keep changing paths once I get a job? And the answers for those are yes, you can do it. You can do it. You can certainly, if you're motivated enough, if you want to change your career path, if you like what you do, you can certainly do it. Will you get a job? Absolutely. I got a job. So you can certainly get a job as well. Actually, is if you go to different types of places, for example, industry, government, people are highly interested in the skills that you have and how you can adapt to different types of project and how you can use knowledge that you acquired previously from different projects into new ones. So those are very valuable skills. So you can certainly get a job. Will people trust the knowledge that you had? Absolutely. If you keep working hard and if you can demonstrate that you can speak to them in a high level that they expect, they will trust your knowledge. But can you keep changing paths? That depends. If it depend on the where you're working on, if it's an industry or government, depending on the mission of the place or the type of scope that they have, you might not be able to have the same academic freedom that you'd have to do whatever research you want if you were in a university. But that depends. So that I cannot really say that it can keep changing on that. If you're unsure about what path would you like to take, what can you do? Well, the first thing, talk to your supervisor, talk to your academic advisor, your professors, anyone that you think could give you a good advice and like what path you should take. And my suggestion would be, maybe you like a lot of things and you don't know where to go. Well, try to go the black side way. Try to eliminate what you do not like first. And then once you have those out, then you have a less amount of options to work with. And then you can talk to them and see, would you suggest something that I could pursue that would align better with what I want? Also, if you are inclined to do research, try to join a research group. Try to do summer internships. Those are always very helpful. They not only expose you to different types of research, to different type of people that might be your first line of networking because you're working with them, but those research groups and the internships, they can become a bridge for you to find a job in the future. That's how it worked for me. I did internships, and that was super helpful for me to find my current job at the agency. If you can attend conferences, professional conferences, career fairs, become members in associations and professional societies. Try to do it. There are a lot of societies for pretty much any topic, scientific related, and usually the annual fees for students are, don't quote me exactly on this, but I don't know the price for all of them, but can be $25 to $35 a year, and you have access to all their conferences, all their career development paths. They have seminars. They can have seminars teaching you how to create a resume, a CV that is tailored for different types of jobs. If you want to go to government, academia, if you want to go to industry, how to tailor each one of them. You cannot write the same resume for everybody. You have access to professionals in the field. You can expand your networking. And those are great ways to start doing networking, as well as LinkedIn. You might have heard that LinkedIn is a great place that you can always reach out to someone and, hey, I saw that you work in such place, you do that type of job. Would you mind giving me some suggestions on how to enter the field or how you did? Usually people are very open for this type of information. And since we talk about internships, I would like to give you some information about that. There's always internships. In this case, I'll present for government. That's my area. But if you are interested in internship opportunities, for example, at the FDA, if you go to the fda.gov and you search for internships and then scientific internships, fellowships, trainees, and non-US citizens, you find a page like this. And then here you have a much longer page that you have information about opportunities within the FDA, in different locations for different types of research that might be of interest, not only related to food, but also related to toxicology, drugs, anything that's on the scope of the FDA. What if you don't want to work for the FDA, you want to work for another government agency? Can always go to USA Jobs. You go in the link here for help, and then work for working government. And then there are hiring paths, students and recent graduates. There are a lot of opportunities for students and recent graduates that you might be eligible for federal internships and job opportunities still as a student or a recent graduate. And you can apply for that in any government agencies. So you just need to check the government agencies, the location, and go for it and see how it goes. So I hope that with sharing the experience that I had throughout my career, I hope that I could give you a little bit more information on this path. If you are someone like me that would like to follow this non-linear career path, and just wanted to give some credits to some of the figures that I used. And with that, thank you very much for your attention, and I'm open to any questions that you might have.
Thank you very much, Dr. Matos. It's very informational across multiple subjects. So I can start with the Q&A, but I'll mention to any students, if you do have questions for Dr. Matos, please put them into the chat now, and then I will relay those questions to her. So while we wait for maybe some students to enter in their questions, I have a couple. You mentioned that you had to adjust, right? When you went from one topic to another, or one field to another, there was that adjustment period that you had to go through in learning the new information and the new research, perhaps the new techniques. Could you talk maybe a little bit more about, how you went through those adjustment periods, and also a little bit of how you overcame, you mentioned the imposter syndrome associated with entering into a new field. Can you talk a little bit about those adjustment periods and how you kind of overcame that possible imposter syndrome that you may have felt?
Yes. Okay, so for the first question, how to adapt to the new field. I always try to do something very focused. So for example, when I was transitioning from plant pharmacology to forensics, when I joined a new research group, instead of trying to learn everything about that field, everything about forensics, you go focused on what is the work that is being done here in this group. What are the interests of this group? Or if, let's say, if you're interested in a job, if you're working, you are changing from food safety and are working in a pharmaceutical company on the opposite side. What do these people want? You don't have to learn the entire field at once. You have to focus on what they are requiring of you. So look for scientific papers, look for books that give you the basics on that, and ask your colleagues also, like, what do you think that I should know on this field to succeed, at least to start? And people are usually very helpful with that too. But if you can, let's say if you join a new group or a new company, you can always look at the type of products that they sell, look at the type of scientific products that they display in their websites, or if it's a research group, look at the type of scientific publications that they published in the last years and start with those. And that's how if you target your adaptation period, that is faster for you to follow what's being said, and you don't get desperate so quickly that I don't know anything. How can I follow? Always remember that whatever scientific knowledge you had will be useful. Knowing how to follow the scientific method, know how to perform a technique in the lab, even if it's not the same one, you have that criteria in your mind, so you'll be able to follow. You just need to adapt. In regards to the imposter syndrome, that is something that I still have to deal with that today. It's impossible to remove that. But what I try to do is use that in my advantage. Have that expression that people say, keep your friends close, but keep your enemies even closer. I see that imposter syndrome is my enemy, and I want that close to me. And by that, I mean that if I start having questions, and that sometimes it's usually from the old person, saying, I don't think I can do this. I think people don't trust me. Try to use that to work on yourself and see if maybe if they don't trust me, maybe if I get better on this, they will trust me more. And when you try to work on yourself, um, you start to overcome that feeling that you are not doing enough, that you don't know enough. And sometimes sometimes, if not all the time, it's usually just in our heads. So it's very important when you talk to our supervisors, to our professors or bosses or whoever's in step above, and try to get some feedback from them. When we ask for feedback and they say, am I doing a good job? What do you think I'm doing good? What do you think I could improve? And when that person gives you feedback, you may feel, oh, I'm actually not doing that bad as I was thinking. Maybe I'm just being very harsh on myself, and that's might be a little detrimental. It's easy to say that just forget and don't, you can do it, you don't need to feel imposter, but you not always we can overcome those feelings. So trying to get feedback from people that you trust, from people who are honest with you, who you not sugarcoat, and you actually give constructive feedback, that is very helpful. But try to keep that in your mind, that use that in your advantage. If you think you're not good enough for one thing, try to prove yourself wrong, and that will be very helpful.
Yeah, those mentors and supervisors, the supportive ones, are so important to find along, your everyone's journey. And I think that those feelings, and I think I stole one of the students' questions in the chat. They asked, do you ever still have moments where you feel like that? And I think that's true for many of us, particularly those of us who are somewhat younger and female, and or minority. I mean, these are pressures and that it's hard to get away from sometimes. But I think, as you mentioned, finding those people who are supportive, that's something that's really helped me, both in my current position and previous positions. And something that struck a chord with me when you were talking about your time in Brazil at doing the research there. You mentioned that the couple of people who you're working with weren't as supportive as what you had hoped they would be. But you did mention that you still found individuals there that wrote you letters to support your application to West Virginia and through your program. Maybe could you talk about how you have been able to identify those supportive types of mentors along your professional journey?
Yes. So as you mentioned, very, very, it was perfect that unfortunately we don't always face the supportive people, and it's very detrimental when we end up facing those not supportive people as our advisors, people who should give us support and guidance. And that's what I had. So my advisor at the time, even though he was not supportive, he gave me a recommendation letter. It was probably the worst recommendation letter among the ones that I had, because he really didn't believe that I would pass or that I'll be accepted, because he was just not supportive at all. The way that I identified people who could support for recommendation letters at the time were, I went for professors that I performed well in classes, that I thought that I had a clear communication path with them, that they seemed to like to give feedback, and they saw some potential in myself. So I talked to them, I explained the situation and said, I really want to do this. I know that's hard, but I don't have a lot of support from my advisor. Would you be willing to provide a recommendation letter based on what you know of me? And they were very supportive, those ones. So in the end, the recommendation from my advisor didn't really hold much of a weight compared to the other ones that knew me from other types of work. Outside of academia, if you're a student, and how to usually find that support everywhere else, I think it's just based on the networking connections. when you talk to people, when you go to conferences, people are usually willing to help, and you can know immediately if it's someone that can be part of your networking, can be helpful. When you attend places, and sometimes we almost create idols when you have research and you say this person is such a famous important name in the field, and you go to a conference and you see them and you might ask for a recommendation and you expect they react very welcoming in a very welcoming way, and they don't feel frustrated about that. That's not always, that do not define who you are or who they are. Just move towards someone else that might be receptive. And I had encounters of other times that I needed recommendations for job applications, and were from people I got from people that I met in conferences, or that I had worked with, or that I had attended a seminar. Hey, I attended a seminar for you. You work with that. I work with something similar. Or I'm very interested in something similar. Would you be able to discuss that? And if you know a little bit of myself and my work, would you be able to give a recommendation letter, or maybe give me a guidance on how to get one? And that always worked. It was easier than what I thought that would be.
Sometimes the hardest part is approaching the individual, I think.
I believe so.
Right. I think more times than not, you're pleasantly surprised in the response that you get when you approach someone. Like as you said, but for at least in my experience, sometimes the hardest part is just mustering up the courage to approach someone, which is why I like to tell my students, you know, if you do have a connection with a professor, don't be afraid to approach them to ask for help for one thing or another, including a letter of recommendation. Okay, so we do have a question from a student who asks if you could mention the name of the plant again, the one that you worked on in your plant biochemistry days.
Is it in the chat? Let me see here.
I think it was a direct message to me.
Oh, okay. The popular name is Janaguba. Let me see if I can type in the chat here. Is the species. It's usually is a popular plant there, medicinal plant, and I had even family who had requests, can you bring this to the US when you go? Unfortunately not, because of the borders control. You cannot bring type of plant natural products, but the research is still going on there to not only be able to create some sort of medication with the plant, but also be able to maintain the ecology and the preservation of the species. Otherwise, people just start cutting the tree and removing all the latex, and you destroy the ecosystem. So it'd be a combination of both. But yeah, that's the name of them, and from previous experiences, that works.
Right. I do just want to mention to the students, I put a link for a survey. So if those students, if you could just take 2 minutes to answer the questions on that survey, we'd really appreciate it. It does help us apply for and maintain funding for the program. And then another related to what you just mentioned, a student mentioned that she's interested in medicine, but also in holistic homeopathic medicine. So she feels somewhat conflicted, although I think your story almost shows how those two can go hand in hand. I don't know if you want to add anything to that.
Yes, so I pretty much believe in the popular medicine. We have a lot of cases that you can prove that that's correct, and including my own research, that you can go with that way. I'm a little bit more skeptical with the homeopathic medicine. You always should be skeptical that when something comes from a plant, it does not mean that it's always work or be medicinal. The plants are not interested in producing anything to human benefits. They are producing things to save them from pests and to defend them from the problems that they'll have in the environment. So you have to take this a little bit with a grain of salt, that even though there is a lot of scientific research related to plants, and we have found a lot of good results as the ones that I presented, Be careful when you follow a research path that is based on things that might not be exactly scientific. They might use some scientific terms. They might use a little bit of scientific knowledge, but they might guide you towards something that is a pseudoscience. As for example, homeopathic medicine is not considered yet a real science, and the holistic approach can to be careful with how you use that. If everything is backed up by science, and my interest was to know, does this plan that everybody takes, is that actually true, that's working? How can I investigate using the scientific method and using methods that can be replicable not only by my lab but from other labs, reproducible, and everybody gets the same results, and then we figure out that yes, we are getting the same results and that can work. How can you use that towards medication? That's one thing. Another thing is when you start diluting products and still claiming that they have the same benefits. So I don't know if I answer exactly the question or the comment, but it's and I know that it's frustrating when you're dealing with a disease, a terminal disease, that you might try to reach to everything. It's what my grandma did. In her case, it worked, and in her case, the plant was powerful enough, the compound, to cure her. But it's something that when you're following research, you have to be careful and do a little bit more digging to see if that is something legit.
Yeah, so I think that I think it was clear things up pretty well, and yes, we often hear those, stories of some type of miracle cure, but as you mentioned, it is important to understand what's really happening scientifically, and part of that is understanding, are there published results on it, and similar to what you published regarding that particular plant. I mean, is there scientific backing behind that particular so-called cure?
The claims, yeah.
I don't see any other questions. Let me make sure I didn't skip anything. I don't see any other particular questions in the chat. So I think with that, then we will conclude our first WISE seminar. And thank you again very much for taking the time to talk to our students today. It was really informative, I mean, for me as well as hopefully for them. And also we had some faculty and staff here as well. So thank you to the students for coming. Again, please do complete that survey, and hopefully I'll see you guys in April when we will host our next seminar in person.
Thank you, everyone. Have a great day.
Okay. Thank you very much.
Thank you.